My name is Beyrak toydemir. I'm medical director in ARUP laboratories and also faculty at the University of Utah. I will talk about molecular genetics. There is no financial disclosure. In this section, I will talk about apply Bayesian calculation to carrier residual risk, molecular test strategies and molecular disorders, and also we'll talk about triplet repeat disease. Cystic fibrosis is an autosomal recessive multisystemic disorder associated with pancreatic insufficiency, malabsorption, malnutrition. It's a progressive pulmonary disease, increased concentration of sweat electrolytes, and an infertility in males. There is also a typical form of CF, is a non-classical, and can present with just one organ involvement. This could be a congenital bilateral absence of vas deferens, CVAVD, or pancreatitis, rhinosinusitis, or nasal polyps. And there is a very wide variation on the expression among the patients, and it could be due to allelic heterogeneity or some modifier genes that we will talk about. Genotype testing is important because it will determine pancreatic insufficiency or pancreatic sufficiency. It will be important because it will predict the severity of the disease, determine required corrective therapies, effect on protein, determines appropriate drug, and also help for the families as well. So this table is really helpful to understand the mutations effect on the cell level and now what kind of therapies will be used for those specific mutations. So I would recommend looking this in great detail. As we all know, cystic fibrosis is common in the Caucasian ethnicities and Ashkenazi Jewish. Here are the incidence for the CF, classical CF, in these different ethnicities and carrier frequencies as well. ACMG and ACOG recommends the carrier screening for CF, and at one point there were only 23 pathogenic CF variants. They were severe or moderate mutations, and this in the populations more than 0.1%, and it was more pan-ethnic panel, but it's more skewed to the European Caucasian descent, and it's been offered to all pregnant females regardless of ethnicity. And these days, some labs offer 23 variant panel or larger panels. It's actually first-tier DNA test for the diagnosis, not for the screening, but that comes with the CFTR full gene analysis, could involve the sequencing and deletions and duplication analysis. Delta 508 is the most common mutation. 70% of all CF pathogenic variants are Delta 508. An ancestral variant, which believed that it's been occurred thousands of years ago, because pancreatic insufficiency. I want to also talk about intron 8 because it's important. There is a poly-T tract and a poly-TG tract next to each other before the splicing of exon 9. And the number of the Ts and TGs are important and affect the splicing efficiencies. If the individual 5T allele is a modifier, but it's associated with decreased levels of CFTR due to aberrant intron 8 splicing. So R117H mutation, if it's on the same chromosome with the 5T, it's act as a moderately pathogenic variant. By itself is actually a mild mutation. So it's important to know the patient if R117H mutation is there, 5T is or not. And then after you find the 5T is there, you need to also find the phasing of the 5Ts with the same chromosome with the R117H or not. 70s and 90s are the benign alleles, and it's been known that the Delta 508 mutation is in linkage disequilibrium with 90. Poly-TG tract could be 11, 12, or 13. And the longer the TG tract, 12 or 13, insists with the shorter poly-T tract, 5T, have the strongest adverse effect on proper intron 8 splicing. For example, patient with the Delta 508 mutation in one chromosome, if the other chromosome is 5T11TG, most individuals will not have clinical symptoms of CF. However, if the patient's one chromosome Delta 508, the other one is 5T12TG or 13TG, this could be a disease-causing variant, and the patients may have elevated sweat chloride, and some symptoms of CFs, or increased risks for male infertility. This is a Bayesian calculation that we always use for carrier risk in this family. What is the risk mean of CF carrier for this individual that's shown in the arrow? And if after the testing of the panel negative, and the sensitivity of the panel is 92%. And here's the answer of this question. Let's look at the pedigrees to find out the prior probability of these individuals just based on the pedigree. So we know the individual 2-1 is affected with CF. That means his parents are carrier for CF. And then individual 2-2 being a carrier, so 2/3 is not 1.5, because we know this individual is not affected. That means that we have only 3 options, and 2/3 is being the carrier. And then the individual being 3-1, the question in the pedigree is a 1/3, half of it. And this is a prior probability. We need to use the conditional probability because sensitivity of this panel is 92%. There's an 8% chance that after the panel, this individual still will be the carrier. And using this prior probability calculations, it turns out it's actually 1 in 26 chance that this individual will be still carrier for CF based on the pedigrees and the panel. CFTR is also included in the expanded carrier screening panels, which you will see the information in detail in this slide. SMA-related spinal muscular atrophies. This is a progressive degenerative disorders. Muscle weakness and muscle atrophy are the main findings of these individuals. Almost all clinical findings are consequences of muscle weakness and atrophy. You can see constipation on the GI, respiratory failure, or scoliosis, all consequences of the muscles weaknesses and atrophy. But however, the metabolic findings are not really well defined. It could be primary or secondary to the underlying the defect. Before the genetic basis of SMA was understood, it was classified into clinical subtypes based on the maximum motor function achieved. However, now we know that actually phenotype of SMA1 associated with SMA spans continuum disorder without the delineation of subtypes. I'm just putting these clinical subtypes for you to read in detail, but I will not go in detail. There are 4 clinical subtypes. Differential diagnosis is important, and there is a disorder called X-linked infantile spinal muscular atrophy. UBA1 gene is the causative gene, or the X chromosome. Another disorder called spinal muscular atrophy with respiratory distress type 1. SMART1 is the type, and the gene is IGHMBP2, important in the differential diagnosis. There are some other neuromuscular disorders, congenital myasthenic syndromes, congenital myopathies should be considered in the differential as well. SMN gene sits on the chromosome 5q. The SMN1 gene is the functional gene, is in the telomere side. SMN2 is in the centromere side of the SMN1 gene. It's important to remember that those 2 genes are extremely homologous in each other. There are only 5 nucleotides that they differ, and none of these differences actually change the onset sequence. And as you can see here, the C on the SMN2 gene in a splice site of the exon 7, it's actually T on the SMN1 gene, which makes the efficiency of the T is important, and it makes the efficiency of the mRNA and include the exon 7, which is important for the function of the protein. So the SMN2 gene, actually 90% of the time, 7 is not in the mRNA, and then 10% of the time it's leaking, which the SMN2 gene is not non-functional, is actually functional, but 10% of the SMN1 gene. And most individuals have 1 copy on each chromosome for SMN1. It could be more copies. SMN2 could be also more than 1 copies as well. 90% of the SMA patients are homozygote for a deletion of exon 7, and 2 to 5% of patients could be compound heterozygote. In 1 chromosome is the SMN1 gene. The other chromosome could have some sequence changes in the gene. And SMN2 gene copy number can modify severity of disease. The higher number of copies of SMN2 actually will have less severe for the disease because it will contribute some protein at the end. Carrier screening has been recommended by ACMG and ECOG as well. And the calculated prior carrier frequency in the United States is 1 in 54, and the detection rate for the dosage analysis is 91%. Individuals 3 or more SMN1 copies will be not a carrier for sure, but if the individual has 2 SMN copies, there is a 5 to 8% chance that these individuals will be carrier because those 2 SMN1 could be on the same chromosome, and it's being called as a silent carrier. It's actually due to the duplication of the SMN1 gene. The individual has 1 SMN copy, which calls carrier. We have to talk about a little bit more silent carriers. It's really important, and it's more frequent in the African Americans of sub-Saharan African heritage or Ashkenazi Jewish descent. There is a variant, it's in the 3' UTR end of SMN1. It's a part of a haplotype associated with SMN1 duplication in silent carriers. Specifically, if the individuals are from Ashkenazi Jewish descent, if they have this variant, it actually increases the likelihood that those 2 copies of SMN1 are on the same chromosome. Approximately 6% of parents of a child with SMA resulting have a normal SMN1 dosage testing. 4% of those carriers have 2 copies on the SMN1 on the single chromosome, but 2% is due to the de novo deletion of exon 7 on the 1 alleles. Look at the techniques to detect for SMN copy numbers. The quantitative PCR is one of the common techniques. You can quantify SMN1 and SMN2 just based on the design, the assays, and the differences between the SMN1 and SMN2, which there are not that many. And MLPA is another technology that's been used widely for copy number changes. And then also there is intragenic variants. However, because those 2 genes are so identical, gene-targeted microarray cannot be used to differentiate the copy numbers. So on the bottom, you can see the example of from the 1 individuals. There is a probe for SMN1 and exon 7. There is a probe in exon 8, and then SMN2 exon 7 probe has been designed for this MLPA assay. And 2 ratio means is 2 copies. So in this individual, there is a 1 copy of the SMN1 gene, and it's been shown in the drop down on the ratio with the red color of the dots. It represents those 2 lower ratio, represents of the 1 copy, and then the rest of them showing the 2 copies. Treatments, Pranza and then Zolgensma are the 2 treatments that are available these days, but the treatments are important to start very early age. The diagnosis in the early age is important before the development starts. Hearing loss. As we all know, hearing loss is a heterogeneous disorder, and 50% of deafness is due to genetic factors. 30% of them are syndromic, and you can see the list of some example syndromes for this. All those individuals will have some other clinical findings other than the deafness. 70% of the time actually is non-syndromic. Patient just comes with the deafness, and 70% of those are recessive. I wanted to talk about this loci naming. It's based on the mode of inheritance. DFN represents deafness, and if it's a DFN-A, it's a dominant inheritance. DFN-B represents the recessive inheritance of the deafness, and if it's being called on DFN, that means it's X-linked. DFN-B is an autosomal recessive form of the non-syndromic hearing loss. Most commonly is GJB2 variants. Is a compound or homozygote cause this DFN-B. It's a 99% of the time. However, there is a 1% of the time it's actually digenic inheritance. 1 mutation on the GJB2 gene and 1 mutation on the GJB2 GJB6 deletion. This is always deletion. It's been described that these deletions actually involving the GJB2 cis-regulatory elements, and that's the reason that's causing the hearing loss. DFNA3 is an autosomal dominant non-syndromic hearing loss. It's extremely rare. So far, only 14 mutations been described. 90% of them are on the GJB2 gene, 10% on the GJB6 gene. Let's talk a little bit more about GJB2. The protein name is connexin 26, and usually progressive, moderate to profound hearing loss. And 50% of individuals with this hearing loss could have actually mutation 1 or 2 pathogenic variants in the GJB2 gene. And this is a very small gene. It only have 2 exons, and there are more than 200 variants been described. However, 35-DALG is the most common mutation. The frame shift creates a premature termination. And the carrier frequency of this allele is 3 to 4% in Caucasian, and it accounts 75% of the non-syndromic hearing loss. So it used to be a TR1 screening, and usually after this negative goes to the next steps. However, these days is actually not necessary to do that. As you can see, this is the region of the 6 Gs and the control, and then these individuals on the bottom actually losing the Gs in the both chromosomes. That's a total of 5 Gs in both alleles. And in the right panel, you can see the heterozygosities. One allele is the 6G, other one is the 5G, and it's a heterozygote deletion. Alpha thalassemia. Alpha gene cluster locates on the chromosome 16p. A normal individual has 4 alpha genes. 85% of those mutations are actually on either partial or total gene deletions, and 3.7 and 4.2 are the most common deletions, which I'll talk about a bit more. 15% of the mutations are sequence variants, and so far more than 100 pathogenic variants has been described. 3.7 deletion is one of the most common deletion. And here on the bottom, you can see the 3.7 deletion cuts half of the alpha-1 gene and half of the alpha-2 gene, and then creates a fusion alpha gene. Alpha-1 and alpha-2 is also very homologous genes, as actually the sequences are same. And this fusion alpha is functional gene. That's why this individual, if he has a 3.7 deletion, this individual losing only 1 alpha gene and has 3 functional alpha. 4.2 deletion only deletes the alpha-2 gene, and alpha-1 is intact. That's why heterozygote 4.2 deletion individuals has only 3 alphas, not 2 alphas. However, there are larger deletions than I listed here, South Asian and 20.5 kbs. Those deletions are extremely large, and it actually deletes both alpha-2 and alpha-1. The carriers of those deletions, the individuals will have only 2 alpha in 1 chromosome. This table is helpful to understand the total genotyping and phenotypical informations. And if you have a 3 alpha, you will have asymptomatic or mild microcytosis. If you have 2 alpha, 1 chromosome could be 2 alpha, the other one no alpha, or each 1 alpha, it's called alpha thalassemia trait. And these individuals will have mild anemia and microcytosis. If individual has 1 alpha, called HBH disease, and moderate to severe hemolytic anemia or hepatosplenomegaly are the phenotypes. And the parts there is no alpha, and this actually is very lethal, and hydrops fetalis is one of the most common finding. This is another way to show all these details that I just talked about. About 15% of alpha thalassemia is due to point mutations, and sometimes they can cause more severe thalassemia than gene deletions. Actually, hemoglobin constant spring is the most common point mutation. It creates a stop codon, results in large alpha globin protein, RNA unstable, and little protein is made. Alpha thalassemia testing methodologies could be testing just the targeted deletions that are just PCR and gel electrophoresis, or for the sequencing of those genes to detect the sequence changes, and then deletion duplications can be detected by MLPA technology, which I'll explain later in my talk. Beta thalassemia, it's the beta globin gene locates on chromosome 11. And there are many variants in controlled regions or splicing defects. It's been described more than 200 known pathogenic variants so far. Opposite of the alpha thalassemia, large deletions are rare for the beta thalassemia. It's similar risk populations, Mediterranean, Middle East, Africa, and South Asia. Beta 0 means no synthesis, beta plus means reduced synthesis. And then you can see the beta thalassemia minor or beta thalassemia trait, and those are mild anemia and asymptomatic individuals. The individuals who have the symptoms is the thalassemia major, and those are really the severity dependent on degree of imbalance between alpha and beta and amount of unassembled alpha globulin protein precipitant. And these individuals will have risk of iron overload and major organ involvement as well. Those are the most common variants in the beta globulin gene. And hemoglobin S is the most common one. I put the most, both nomenclature. The nomenclature has been used many years, and then the new nomenclature, there is differences between, because at the beginning, the metion at the beginning, it wasn't being counted on the beta globulin protein. And those abnormal beta globulin proteins can be identified by HPLC or IEF, and the testing technologies are similar, could be sequencing or MLPA to detect the large deletions. Factor V Leiden mutation, it's a missense change, arginine 546 glutamine, and in gain of function missense variant, and it's autosomal dominant inheritance with the reduced penetrance. These individuals have higher risk for developing venous thromboembolism. You can see all the different risks and the clinical phenotype based on using the contraceptives or pregnancies and some other factors that it may involve and increase the risks for these individuals. The detection of the factor V Leiden could be very simple and very economical ways using the melting curve analysis. In this technology, we use the melting curve analysis. We use PCR primers and the probes in the same mixture after the PCR. And probe binds the increased temperatures after melting, after the PCR, the melting, and the probe melts away. And then based on the matching probe, perfect match with normal allele will melt later in the higher temperature. Mismatches will actually melt in the lower temperature. In this example, as you can see, the blue represents of the homozygous mutant, and then the higher temperature, the black line represents the normal, because our design the perfect match with the normal allele. And then the red shows the heterozygote peaks, and the mismatch, the homozygote allele actually melts. Sometimes even you see this unexpected peaks can be seen. In this example, the pink is the wild type allele and blue peak in the lower temperature is the mutant allele. However, in this example, there is a green peak doesn't match with the heterozygote, but one peak doesn't match with any of the peaks. The other higher temperature peaks match with the wild type allele, so that means it's something else. There's another mutation or polymorphism exists, and the only way to know this is just looking at the sequence to identify if there is another mutation or polymorphism other than the we're looking for, because this is a targeted testing. In this example, we saw that very next nucleotide of the 1691 is our Leiden mutation. In this example, there is a variant on the 1690. Prothrombin is a factor 2 autosomal dominant inheritance with a reduced penetrance. And this variant exists on the 3' UTR end. The naming is 2021O G2A. It's a gain of function variant and increases stability of prothrombin mRNA. This way, increased levels of prothrombin results in increased level of thrombin and then increased clot formation. If it's homozygote, increased thromboembolism 3 times compared to the heterozygotes. Detection of this targeted mutation could be the same, like I talked about factor 5, could be melting curve analysis on the right corner. And it could be some other technologies as well. One of them is in the right on the bottom of the corner. It's really just the PCR restriction enzyme, just to able to identify or differentiate the mutation, and then the gel electrophoresis and old school technique. It could be really complicated in a way, but it's more sophisticated and can actually quantify at the same time. In this assay, you can use a TAGMAN assay, and you can design 2 probes for each location, and then each of them could be labeled, and they are labeled, and when they hybridize and they ligate and merization, there will be a signal, and then you will get the result from that allele. So if you have 2 probes for each mutation, you will have a different signals and the different colors. Let's say FAM represents of the A, and then the WIC represents of the G. And then individuals only have the G allele, will not have the FAM allele. So you will only get a higher amount of the quantification of the Gs. And then if the patients or the samples are heterozygote, you will get the amplification of the both alleles. And then there will be amount of the amplifications will be in the middle of the both. You can see the same sample with the amplification curve, heterozygous in the middle, the G to A is actually the cycles are here, that, and the yellow represents the no amplification, and then orange represents the higher amplification for that mutation, that the homozygote wild type. Achondroplasia is the most common form of inherited disproportionate short stature. And as you can see, the clinical findings, macrocephaly, like bovine and many others. And FGFR3 is responsible. It's a gain of function hotspot variants, 100% penetrance. And there are only 2 common mutations as on the same nucleotide in same amino acid position. One is G2A, is a 98% of the time, and G2C 1% of the time. 80% of those mutations are de novo mutation. We can still detect these mutations using the melting curve analysis. And pink represents the wild type allele. That means our probe is perfectly matched with the wild type. And in the higher temperature, as you can see, the yellow represents the C allele, and then the blue represents the A allele, which actually melts in slightly different temperature. There are other types of the skeletal dysplasia. It's coming the tonotropic dysplasia called TD, and those are mostly lethal in neonatal period, and most of the cause are 3 variants, and it's been divided in 2 types, TD type 1 and TD type 2. TD type 1 is the curved femur, flat vertebral bodies, and multiple molecular defects, but this arginine 248 cysteine is the most common one. And TD2 is a straight femur, tall vertebral bodies, cloverleaf skull, and single causative molecular defect, as you can see in the FGFR3. Here are the most common mutations on the TD1, TD2 gene, achondroplasia gene, and hypochondroplasia disorders, and all of them are on the FGFR3 gene. And you can see the positions of those mutations and locations in the gene as well. And since there is not only 1 or 2 mutations, more than several mutations needs to be detected. Melting curve analysis may be not a good technique to do that. There is another technology that I wanted to introduce to you, single nucleotide extension technique. You can actually detect up to 10, 12 mutations easily with using these technologies. Maybe mini sequencing, because you anneal your primer adjusts to the SNP base. Here, the red represents your primer, differentiate the allele, and then after you add polymerase and DNTPs, then after you do the mini sequencing to able to identify the mutation, and also mutation has been identified by the capillary gel electrophorezis with the sizes as well. You can see it, even if it look very messy here, that each peak represents of the 1 mutation, and then in this panel 9 mutation has been targeted. In this individual, there is a 1 wild type allele T, other one is the G is a mutant allele, and we can detect both of them is actually positive for the TD1. I'm just gonna switch to the X-linked inheritance, and as you can see in the pedigrees, X-linked recessive are the females are carriers, males are affected, and the dominant males are mostly can be fetal, the females are affected. And one of the most common examples of these disorders are the dystrophinopathies. Dystrophinopathies could be ranging from mild to severe, and DMD, the Duchenne, BMD for the Becker, and also the dilated cardiomyopathy, DCM, are the most common forms. Duchenne muscular dystrophy is the most severe form, and individuals wheelchair dependent before age 13. And Becker is the more light form of this disorder. Individuals wheelchair dependency is usually after age 16, although some individuals can remain ambulatory in their 30s, and preservation of neck flexor muscle strength is important for differentiation BMD from the DMD. The associated dilated cardiomyopathy is predominantly dilated cardiomyopathy with left ventricle enlarged and weakened. And those individuals usually presenting between ages 20 and 40s, there is no clinical evidence of skeletal muscle disease, only on the cardiac muscles. DMD gene locates on the XP21.2, and it's really large gene. And there are lots of tissue-specific promoters and splice variations. It's important for the developing different type of disorders. The most common mutations are the deletions. 60 to 70% of the time are deletions. 5 to 10 of those mutations are duplications, which encompassing one or more exons. And there are 25 to 35% of the time small insertions, deletions, or point alterations, which can be detected by sequencing. 2/3 of the cases inherit their mutations from their mother. And although multiple technologies can be used, MLP is the most common to detect the deletions and duplications, and NGS is for the sequence change because it's a large gene. BMD is a pathogenic variants that do not cause, BMD is the milder form, and then the pathogenic variants that do not alter the reading frame cause most of the BMDs. And if the pathogenic variants alter the reading frames, the out-of-frame mutations or very large deletions, it actually causes a DMD. And type of the deletions or duplication depends on the out-of-frame or in-frame, can be diagnosed DMD or BMD 90% accuracy. And most common deletions are involving exon 45 in between 45 and 55. And DCM, the cardiomyopathy form, is actually the pathogenic variants are in the DMD, but affect the muscle promoter and then first exon of the dystrophin, which express on the cardiac muscle. There are some other isoforms mutations been reported, and these individuals develop intellectual disabilities. So the isoforms are important for developing the different forms of the disease. This is just showing where the VMDs or DMD deletions are located in the dystrophin gene. And I wanted to give a little background about the MLP I've been talking about during my talk. MLP is a multiple ligation dependent probe amplification technique. And it's a semi-quantitative technique that can detect deletions or duplications on the region. Each region that you wanted to quantify, you need to have two pieces of the probe, and those blue lines represents the perfect match with your sequence that you wanted to quantify, and green part is just a stuffer sequence that differentiate all these different pieces of the probes. And X and Y's are the PCR primer sequences, but they don't match with any of the human genome. This way, you can only amplify these probes that hybridize with your sequence. And it's a multiple ligation dependent probe amplification. You can actually do this for many of the regions at the same time. You can develop like a multiplex reactions up to 50, 60 regions. After your probes hybridize with your region, let's say exon 46 and exon 47 here, and then after the hybridization, there is a hybridization step, and then comes with the ligation, the ligase enzyme, if it's a perfectly hybridized and works perfectly, then now we have a one piece of the fragment that you actually do amplify, and then after the amplification, this green stuffer sequences creates the differences on the size that when you do capillary electrophoresis, you can actually differentiate them. You can see the examples. So top is the control, the internal control, and on bottom is the case result. If you look at the exon 13, the peak height, and then exon 13, the peak height, your sample is lower when you use many controls and relative controls, and you can actually see that's actually 1 copy instead of 2 copies. Here's an example from one DMD case as a male. And in this example, exons 5, 6, and 7 has been deleted. On the left side, you can see there is no copies of, because the male, only 1 X chromosome, and it's been deleted. There is no hybridization or no copies. And then you can see it's 3 red dots on the bottom of this graph. And this is a really large gene, and that's why you have to use more than 1 multiplex reactions. And this panel shows the 1 multiplex reaction represents the plot here. And then the second panel represents the second plot with the plot over here. The ratio is over here. As you can see, the 0.7 is our borderline, and this green dot represents 0.727, which is in the borderline. It could be deletion or something else. And this is one of the disadvantages or downside of the MLPA, or sometimes can use as advantages as well. However, if there is any mismatch on the probe binding site, this ligation may not work. Depends on where the mismatch is. If the mismatch is just on the ligation, totally may not be ligated, and you won't get any product. It's shown here. Or if the mismatch is a little further from the ligation, can still work, but not efficiently. If you remember from the previous slides, you can have it at 0.7. It's maybe the ligation efficiency is not good, and maybe there is a polymorphism close to the binding site. If there is no ligation, that means there is a change on the mismatch. There is a change. If you sequence that, you can find it. There is a polymorphism, or there is a different mutation that affects the binding site that could actually cause a false positivity of this technique, as you can see in this slide. Before I finish the DMD, I wanted to talk about a little bit for the Bayesian calculation based on the pedigrees. What is the carrier risk for this individual 3-5 shown in the red arrow? And as you can see, the individual is affected with DMD, and his daughter will be a carrier. And then there's a 50% chance for her daughter to be carrier. But what about in this pedigree? What is the chance individual 3-5 being a carrier? To be able to do that, we need to understand what is the risk for individual 2-2 here, another arrow showing what is the risk being carrier for this individual first. Based on the pedigrees, since this individual is affected, and then that means that his mom is carrier, and then, however, we don't know if this individual will be carrier or not. There is a 50% chance. And let's say this is a prior probability to being this individual is a carrier, and it's a 50%. However, we know more information for this individual because she has 4 unaffected individuals, males, which it's more than just a 50% chance. Then we have to use this information in the Bayesian conditional probability as well. So being in those 4 unaffected individuals, you need to multiply this 1.5 4 times because of the 4 males, and there is a conditional probability is 1/16. And then if you calculate this joint probability, the chance of being this individual 2-2 is a 6%. And then being a carrier risk for this individual, it will be half of that 3%. I want to switch gears to the expansion and triplet disorders. And those repeats, the expansion of the repeat disorders could be in the protein coding sequences, which can cause a toxic proteins, or repeats in the RNA only coding regions and could change out RNA function, or in the non-coding regions can cause reduced transcription or translation. And there are so many of the repeat disorders that there's no way that we can go over. However, I'm going to cover a couple of them. This is a good summary of the locations and type of the repeats of those disorders for you to study. Repeat diseases, they actually do have multiple inheritance patterns and could be dominant, recessive, or X-linked. And diseases associated with the expansion and repeat number may not be accurately determined by the NGS assays, so it really requires a specific design to able to detect those expansions. Fragile X is the most common one. The FMR1 gene encodes FMR protein, and this protein expressed in highs in brain, important in embryonic development. CGG trinucleotide repeats locates on the 5' UTR of exon 1 on the FMR1 gene, and there is AGG interspersed repeats is important as well. So allelic diversity results in number and length of repeats and number and location of interspersed AGG repeats as well. And if the CGG expansion gets long, and then there is an abnormal methylation of DNA, this causes deacetylation of histone proteins and gene silencing. However, there is a premutation syndrome, which is actually gain of function mutation. It's a toxicity of the mRNA, which I'll talk about in a minute. This is the phenotypes and physical and behavioral findings of the Fragile X syndrome and the population frequencies. But I want to really focus on the Fragile X premutation phenotypes. It's actually unique disease entities caused by overexpressed elongated mRNA with a gain of function properties. If this permutation is in the males, cause tremor ataxia syndrome. And it's a cerebellar gait ataxia, action tremor, Parkinsonism. If the permutation occurs in the female, it causes primary ovarian insufficiency. And it's causing early menopause on the patients. Actually, females also have a risk, but less chance to develop tremor ataxia syndrome as well. You can see the number of the repeats and then the frequencies and the expression pattern in this table in detail. Premutation female transmission is important, and premutation female carriers has a 50% chance of transmitting unstable allele. And the females transmit the premutation alleles to be extended stage. However, on the males, actually unlikely to expand to the full mutation. So that's important differences between those two. And gray zone in intermediate alleles is between 45 to 54. Those may expand the premutation allele when transmitted to offspring in some families, but does not expand to the full mutation. And AGG interruptions are important because that's actually stabilize the repeat, reduce the likelihood of the expansion. So sometimes that's why important to know the AGG repeats, but it does not affect the diagnosis of the Fragile X. This is really typical family pedigrees. As you can see, the top generations in this family can come with the Parkinson's type of phenotype or early menopause, but then the next generation can come with intellectual disabilities, which because it's expanded. There's sudden blood technology. It's called gold standard technique to detect this expansion and also the methylation. However, it's not really used anymore because it's really difficult technology to use, but I'm just going to go over the technology just in case if there will be other questions, and it will be good to learn because it still is a gold standard. Technology, you need to use a methylation sensitive enzyme, and there is also a general enzyme like EcoR1 that you want to cut the DNA in the small pieces. And then after you need to cut with the methylation sensitive enzyme to able to differentiate methylated pieces, unmethylated piece. Here's the graph shows the real result from the southern blot. This enzyme cuts when the X is active. And when it cuts, it will be shorter fragment. Here is represented this one fragment is shorter than the other fragment is actually one. This means that this individual has one active X, which is a normal male. And normal female, based on the X inactivation pattern, you will have one active X and one inactive X, and then it will be normal. This is the normal range of those both cut-in and uncut fragments. If you have a pre-mutation, you will have one chromosome will be the normal range because it will be normal repeat number. The other chromosome will have the large enlargement, but based on the random X inactivation, both of them could be active or inactive. So you will have four options: that active X with the small repeat number and large repeat number active, and an inactive representation of the small repeat and large repeat. So this is a pre-mutation female. And here you can see that there is no active X and everything has been methylated under this region of the fragile X, and it's been enlarged from the regular sizes. So this is a full male. There are other easier techniques being developed last 5-6 years that actually most of the time the labs uses triplet repeat prime PCR. In this technology, you have the both primers. However, actually it's just a regular primer, but then the reverse primer, you can add the CGG repeat in the primer. This creates each time that the reverse primer binds, if it's an enlargement, and you start seeing this starter peaks in here in this kind of a pattern. This pattern represents that there is an enlargement of the fragment. The reason that using triplet prime PCR, because if you do regular PCR, just look at the size, the enlargement of the full mutations are really big. It's usually 1,000 repeats, and then the PCR cannot amplify the large allele. So then you will actually have an allele dropout of the large allele, and you will not know that there is an enlargement of the of the patient. So that's why you need to have a special trick to able to detect this enlargement allele. Even if you cannot PCR the whole fragment, you can see the pattern to able to detect that this individual has enlargement. As you can see, the female, it's not expanded, it's just almost one peak and there is no enlargement here. And then in this example, one allele is normal, the other allele is enlarged. You can see one allele is big, but then it's continuous with the very long 200 phases of continuum of this started with this reverse primer, creates this starter position. And in here in male expanded, there is no normal allele. Everything has been the starter positive peak. And on the bottom, you can see normal female, the 29 and 39. There is nothing, it's been varied. On the left side, the full mutation female, you can see one allele is in the 39. And then almost there is not second allele. When you zoom in, you can see this pattern that it represents that this individual actually has a second allele, which totally has been enlarged. But you will not know with this technology that's actually been methylated or not. You need to have a methylation sensitive technique to be able to differentiate the methylation. This individual has one enlarged allele. And on the right, you can see the 31 and 69. Actually, you can PCR 69, and you can see the top allele here. And then when you zoom in, you can see there is nothing else other than the 69. So that means that this individual is really in just the pre-mutation range female. That's the zoom in the enlargement allele and showing the pattern. It's really consistent, and this probably is a not really real peak. And then it's hard to size the enlarged allele using this technique, but you can see it's been enlarged. As more example, you can see the normal male and normal female with the two peaks. And like I said, AGG genotyping assay you can use. The addition of this AGG interruptions in the CGG repeat decreases the instability of the allele during transmission from parent to child, and it decreases the risk of expansion of pre-mutation allele to full mutation allele. However, it's not useful for the diagnosis of the individuals. Huntington disease is another triplet repeat disorder, and it's a dominant CAG triplet repeat disease, and it's actually the repeat sits on the exon 1 of the Huntington gene. And incidence is 3 to 10 to 100,000 individuals. First manifestations are usually neurological manifestation, but some psychiatric manifestations can happen as well. This is a late onset disorder usually, and the repeat numbers expansion actually indicates that larger the expansion, earlier the findings of the mean age of onset. Here's the example of the one pedigree. This individuals, the father of these several kids, the age of onset at 45. And then when you look at the kids, two of them are affected and in earlier age, because this expansion actually occurs in the male gametes. If the affected individual is male in the family, next generation could start disease earlier age. Inverse correlation between age of onset and CAG repeat number. And there are some modifier and environmental effects also affect the age of onset. That's important to keep in your mind. And this protein is actually expressed in all tissues. This table represents the repeat numbers and how it's been called. If it's a 40 or more, this individual is called Huntington disease with a complete penetrance. And one of the good methodology to use is a triplet primed PCR, or you can use a fluorescent PCR. But the fluorescent PCR, if you don't create this triplet repeat prime PCR, you can always have a question if there is another allele that may not be detected by this assay. Here, is this individual a 2020, or is this individual has an enlargement? If this individual has an enlargement of the second allele, which the PCR cannot amplify. Mostly these PCRs can amplify up to 50 to 60 repeats. If the individual has 65, it's really important when you design these assays that you really need to know your upper limit of the detection, and then you need to run that repeat number of the controls in your assay. Here, you see the 1819, 1722. When you see the two peaks, is actually you feel more confident that you actually detect. But if you see one peak, and you may actually question if this is a second allele, there is a missing there. But triplet prime expanded assays give a little more confidence on this. For example, in this individual, you can see one peak in 17. So the question is a 1717 or 17 expanded? However, when you look at this pattern, like I showed you on the Fragile X, there is no starter positions or any sign of this enlargement here. So that's really confidently you can say this individual is homozygote. Here's an example of the 19 and 41, which is the affected for Huntington. And you can see that after the 19, there's this pattern that can tell us that the individual has a 41 and stops there, because the pattern also stops there. That's all I have so far, and I'm happy to answer any questions. You can email me anytime, and thank you very much for your time.